Hello everyone. Let us continue with the chapter Sexual Reproduction in Flowering Plants Part 3. In the previous video, we have learnt about the process of microsporogenesis. So what is microsporogenesis? Formation of microspores. So as a result of microsporogenesis, how many pollen grains are formed? 4. So microspore tetrad, you call that as. So here that microspore is nothing but the pollen grain. So in today's class, we are going to learn about the structure of the pollen grain. So this pollen grain, it represents the male gametophyte. Pollen grain represents the male gametophyte. So the pollen grain, it measures about, when you just look at the size, it is about 25 to 50 micrometers in diameter. Okay, and when you just look at the structure of the pollen grain, this pollen grain, it consists of 2 layers, okay, 2 walls. So one is the intine and the other one is the exine. So when you just look at the exine, it is a hard protective outer covering. So the outer layer is called as exine and the inner layer is thin and continuous. So this is the inner layer. It is thin and it is continuous layer, and this layer is called as the intine. So exine, it is hard and a protective layer, and this exine, it consists of Pulanen. Pulanen. So what is the Pulanen? It is a resistant organic material. It is the resistant organic material which has the tendency to withstand high temperature, strong acids and alkali. That is very, very important point to be noted. It can withstand. Okay, so this Pulanen, if it is present, it can withstand high temperature and then strong acids and alkali. So this is the main important function of this Pulanen. And when you look at this structure, in some places, this is absent. Okay, so in some places, so I'll just mark it here. Okay, so this is the exine layer, what I've drawn here. So if I just give the break here, okay, so likewise here. So here there is a break. So here this region, okay, you call this to be germ pore. Germ pore. So in these places, the Pulanen is absent. Pulanen is absent in germ pores. Okay, so pollen grain, this is the structure of the pollen grain. Okay, it is protected by the intine and the exine. The outer layer is the exine, which is hard. Okay, and here this outer layer, it is made up of a substance called as Pulanen. And in some places, here this is absent, the layer. Okay, that region you call that to be germ pore. So in germ pore, Pulanen will be absent. Right. And next one important thing what you have to remember about this Pulanen is there are no enzymes to degrade the Pulanen. So till now it is not found out. Okay, so that is the main thing you have to remember about the Pulanen, right? And next one, the inner layer is called as intine, which is thin and continuous layer. And this intine is made up of a substance called as cellulose and pectin. Right? So intine, it consists of substance called as cellulose and pectin, and exine consists of substance called as Pulanen. And one more important thing you have to remember about this Pulanen is because of this Pulanen, they are preserved as fossils. Pollen grain, it is preserved as fossils only because of this Pulanen. Okay, right. Now, when you just look at this structure, right? So this is a cell which consists of a nucleus and a cytoplasm. If you take, this is the nucleus and it has cytoplasm. So when a cell undergoes division, so what type of division takes place here? Mitotic division takes place. So what happens during mitotic division? What is mitotic division? It is equational division. So during mitotic division, the nucleus starts dividing. So followed by, what is the division of nucleus called as karyokinesis, followed by cytokinesis. So here, when the nucleus divides, cytoplasm also divides. In that case, when the nucleus divides, it gets separated here, but it is not divided equally. So what happens here? You can see here a large nucleus, okay, and a smaller nucleus present towards the corner. So in this region, actually, here you can see the spindle shaped. Okay, and here this portion, the larger portion is called as a vegetative cell. Larger portion is called as vegetative cell, and smaller one, this portion is called as a generative cell. Okay, and here when you look at the vegetative cell, it has irregular shaped nucleus. Okay, and also cytoplasm is present. Likewise here generative cell also has a nucleus and a cytoplasm. But when you just look at the generative cell, it floats, it is seen floating inside the cytoplasm of the vegetative cell. Okay, when the pollen grains are released, we have seen in microsporogenesis, formation of microspores. When the dehiscence take place, when the pollen grains are released, they are shed at 2-cell stage in 60% of the angiosperms normally. Okay, it is shed in the form of 2-cell stage. So now this portion, you call that to be what? 2-cell stage. Okay, so the pollen grains are shed when it is 2-cell stage. Okay, in most of the, where when you talk about 60% of the angiosperms. Okay, then what happens in remaining species? It is shed in the form of 3-celled stage. So what is that 3-cell stage? In some cases, this generative cell, it divides into two. It divides into two. So the nucleus, okay, you call it as a gametes. So here it divides into two. So totally it will become 3-celled. Okay, so when the pollen grains are shed as 3-cell stage, it is remaining of the other species, right? Here it is 60% of the angiosperm, it is shed as 2-cell stage. So in remaining species, it is shed as 3-celled stage. Okay, so that is why here this pollen grain is referred to as the male gamete. Then when you talk about the importance of the male gamete, male gametophyte, that is the pollen grain. This pollen grain, as it is very specialized in having the nutritive material, okay, this is used as a supplement, food supplement, right, in the form of syrups, tablets. So that is very important thing where you have to note down this. And one more thing, even this pollen grain, it is taken by the athletes to perform performance of athletes and even race horses. Okay, and in some cases, it also results in allergies. It causes allergies. It brings about bronchial afflictions in some individuals, where it may lead to bronchitis, asthma. So these are the respiratory disorders which is caused due to the pollen grain. So one way if you take, it is also beneficial. The other way if you take, it is also harmful, which causes allergies even in human beings, which results in these cases, bronchitis and asthma. Likewise, very good example what you have to learn, remember here is Parthenium. Parthenium you would have heard, right? Which is introduced in India as a contaminant along with the wheat species, and this is also called as carrot grass. Parthenium or carrot grass. Even the pollen grain, it causes allergies. Right? Okay. The one more thing what I have to learn, whether the pollen grains have viability before the fertilization occurs, whether when the pollen grains are released, whether it reaches the stigma within the correct timing, whether the fertilization take place, what about the viability? That is very important thing what to be noted. Then example if you take like rice and wheat, Example, rice and wheat. Okay, so the viability of pollen grain, it can lose its viability within 30 minutes. So within 30 minutes it has to reach the stigma and the fertilization have to take place. Okay, but if it does not reach the stigma within 30 minutes, it will lose its viability. So here within 30 minutes it will lose its viability. That is one important thing. So the pollen grain lose viability within 30 minutes. But in some examples like Rosaceae families, Rosaceae, then Leguminosae, okay, then Solanaceae. In all these species, the viability remains even in months. Viability remains even for months, right? And you would have heard about the cryopreservation methods. Even these pollen grains can be preserved for many years. Even semen of many species, like human beings also, it can be preserved for many years. And how it can be preserved? It can be preserved, okay? So you call that to be artificial insemination. Okay? So the semen can be preserved in liquid nitrogen, where the temperature is maintained minus 196 degrees Celsius. Right? So likewise, even the pollen grains can also be preserved in liquid nitrogen, minus 196 degree Celsius. Like you call it as pollen banks, and even like seed banks also. Why? Why? Because it can be used in crop breeding programs. That is very, very important. Okay, so in this case, when it is going to be preserved, it can be used in crop breeding programs. Okay, so today's class we have learned about the structure of pollen grain and what are the beneficial activities and how it is harmful and what about the viability, all those things, right? If you like this video, share, comment and subscribe and follow me on Facebook and Instagram. Thank you.